We are back with yet another intellectually engaging episode of Immunology for Everyone podcast by SKA. This episode 6 will address the reasons how the same pathogen is responsible for both acute as well as chronic infections in different individuals. The immune system components are clearly playing a massive game, being impacted by various factors. Dive in here to see what they are and what they do. It gets even more interesting with your questions. Continue to like and share these podcasts, and don't forget to subscribe for alerts.
Hello, everyone, and welcome back. So, in the last few episodes, uh, we have talked about the antigen presentation, and we talked about the dendritic cells being the most potent antigen presenting cells, which are responsible of priming the naive T cells. This process, we know that it involves multiple receptor ligand interactions, which occur between the antigen presenting cell and the T cell. And this particular interaction is called immunological synapse. We also understood that the immature dendritic cells in the peripheral organs, they capture the antigen at those sites, and after processing of the antigen, they present the antigen epitopes displayed on various the one of the two major histocompatibility complex molecules, and the T cells are able to recognize the antigen only when they are displayed in this manner. Then we also discussed that the dendritic cells, they start expressing many other molecules, which are called the co-stimulatory molecules on their surface. And these are very important because the co-stimulatory part on the T cells passes the second signal during the antigen presentation. And this second signal is absolutely mandatory for activation of the interacting T cells. This means that if the second signal is missing, then the dendritic cell will not be able to activate the T cell. So let us consider the circumstances when second signal can be missing and what will be the consequences of that. So it can happen in real life in two situations. One is that if the dendritic cell fails to mature at all, and in that condition, it will not be expressing any co-stimulatory molecules. While the other condition can be that the dendritic cells, they do mature, they do express these molecules, but there are certain negative forces which will suppress this expression, and then the dendritic cell is not capable of passing the activation signal. So one situation when it happens is when dendritic cell captures the self-antigen during the process of development. And in that situation, we know that dendritic cell will not start expressing the co-stimulatory molecules. And that way, they will have the self-antigen displayed on the MHC molecule on their surface, and they will interact with the T cell, which has got a T cell receptor for autoantigens. But that interaction, because it is through only one molecule, the MHC molecule and the TCR interaction, this is a tolerizing signal, and the T cell will become tolerant to the antigen. And this is a good situation because that is the way how dendritic cells help in maintenance of central tolerance against autoantigen, and it prevents us from autoimmune diseases. While in the second situation, it can happen where some pathogens or maybe cancers may exploit this particular property to evade the immune response against them. And let us see what are such conditions. I will try to explain this in context of a viral infection, which is the hepatitis C viral infection, because this viral infection causes acute as well as chronic disease. Clinically, if we look at the situation, about 20 are self-limiting, and because of the self-limiting disease of this infection. However, about 70% of infections, they go on to have a chronic phase, course of disease, which means that the virus is not cleared, sorry, and from the liver cells, and it persists for many, many years. And majority of these people, they go on to have cirrhosis of liver or maybe hepatocellular carcinoma. So now, if we look at the immune profiles of acute and chronic infections, we find that in a individuals who have only acute course of the disease, there is a robust T cell activity. It's a broad spectrum T cell clonal expansion, and a lot of these activated T cells are found in the liver, and they are responsible for clearance of virus specific, virus infected hepatocytes. And that happens in about 2 to 3 months, which leads to the symptoms as well as the resolution of the virus. While in the chronic disease, the literature says that the activation of T cell repertoire is not as robust as seen in the acute, and it is a weak kind of response with narrow range of clonal expansion, and the T are not found in the liver. And that's the way virus persists. This indicates that there is an apparent difference in the availability of activated T cells in the liver between acute and chronic infections, which also correlates with the virus clearance, making it obvious that the availability of activated T cells is very important, and activated T cells play a dominant role in clearance of the virus. So many workers have been trying to understand the basic mechanism of this T cell activation defect, because and many people are trying to work on that aspect. There is one school of thought, including my own lab, which believe that it may be because of defective antigen presentation or defective priming of T cells, so that they are not getting activated. So here I'm going to actually quote some of the findings from my own lab, which are directly associated with these questions related to non-activation of virus-specific T cells in chronic HCV infections. So I have cited some of the references at the end, which you guys can go and read. And these are the papers which have been published from our group in collaboration with leading hepatologist, Professor Yogesh Chawla, and his team. And I acknowledge the contribution made by the clinicians as well as team of research scholars who worked in my lab for many years. So at that time, we hypothesized that the T cells do not get activated in chronic viral hepatitis because these cells do not get proper activation signal from the dendritic cells in this disease. So in order to understand the underlying mechanisms, in my lab, we found that the myeloid dendritic cells, which we could isolate from the chronic hepatitis C patients, and we tried to, the bacterial lipopolysaccharides, and we found that these cells were defective in, expression of these CD80 and 86 molecules, which are the co-stimulating molecules. However, the cells which were isolated from a healthy individual, they were able to really upregulate the expression of all these co-stimulating molecules. So that showed that these dendritic cells from chronic hepatitis C patients, this maturation defect in myeloid dendritic cells of chronic hepatitis C patients was associated with upregulated expression of many negative genes, which included the suppressor of cytokine signaling gene. It's a family of these genes which suppresses the activation of, antigen presenting cells, like indolamine oxygenase or PD1. So all these negative regulatory genes were upregulated. So our data from my lab, it also revealed that the response to antiviral treatment in chronic viral hepatitis C patients was also associated with the maturation status of dendritic cells. So that was the observation which indicated that the dendritic cells carried a maturation defect in chronic viral hepatitis C. And then we thought that whether this particular defect is specific to hepatitis C virus infection or it can happen in other chronic infections. And then we thought of an infection by HIV. And we all know that HIV also causes a chronic disease because the HIV infects CD4 positive cells and it leads to progressive decline in CD4. Takes about 7 to 10 years for a person to develop AIDS when the CD4 count goes below 200 cells per microliter of the blood. And that is the condition where we call it the advanced HIV disease or acquired immune deficiency syndrome. And at that stage, the person becomes severely and the person start getting opportunistic infections. So we wanted to understand if there is any association of immune suppression with functional status of dendritic cells in HIV patients or not. So we investigated the maturation status of myeloid dendritic cells at various stages of disease. In the early stage, when the CD4 counts were near normal, in the intermediary stage, when the CD4 counts were between 250 and 350, and in the advanced stage, when the CD4 count went below. And it was found that the dendritic cells were functioning normal till the disease progressed to advanced stage. And during advanced stage, we found that again, the dendritic cells were unable to mature ex vivo in response to bacterial lipopolysaccharide, which we used as a prototype of pathogen-associated molecule. And we thought that it should upregulate the co-stimulatory molecules. And here again, we found that upregulation of many negative regulatory genes like SOX1, SOX3, SHP1, IDO, all these negative regulatory genes were basically upregulated, and that was associated with dendritic cell maturation defect in these HIV patients who were in the advanced stage. Then to prove this to better, we knocked down the SOX1 gene expression in some of these cells, and we found that there was restoration of dendritic cell maturation capability to some extent, which means that there is a possibility of restoring the antigen presenting capacity of dendritic cells if we are able to block the expression of negative regulators. So based on all these findings from my lab and others, in the literature, we can make an interim conclusion here that some pathogens, especially some viruses, which can cause a chronic infection as they are able to persist in the body, they can manipulate with the maturation capacity of dendritic cells, which can be one of the mechanisms to evade the immune response by these viruses. Cytotoxic T lymphocytes, and that is how these viruses are survive. They are able to survive for longer periods of time in the host. So that is one part. Now let us look at another situation. that is also related to antigen presentation and the necessity of co-stimulatory molecules. In this situation, when the co-stimulatory molecules are actually expressed, but the T cell remain cannot be activated, it is because there are certain other molecules which are expressed by the T cell, which are called which can suppress the T cell activation. There are many such molecules which get upregulated. They are called co-inhibitory molecules. So as we have the activating molecules, we have the co-inhibitory molecules which are expressed on the T cells. And these co-inhibitory molecules, they downregulate the activation process. So one of such molecules which was first to be discovered, its name comes from cytotoxic T lymphocyte antigen 4 because it was discovered on the activated T cells. So its function was thought to be a regulatory function because after activation, it will always try to suppress the activation of T cells. And the more important, interesting observation is that the cytotoxic T, this CTLA4, it interacts with CD80, which is the molecule which is basically responsible for interaction with the molecule CD28, which is expressed on the T cells. And that interaction of CD28 and CD80 passes the second signal. But if CTLA4 is expressed, it does not allow the interaction of CD28 with CD80 because competitively it will inhibit because it has 4 times higher affinity to CD80. So every time CTLA4 is there, it will preferentially bind to CD80, and that will cut down the activation process because it will block the second signal. So this interesting part is that so CTLA4 basically is considered as one of the checkpoint molecules. There is another one which is called the PD1, and this was earlier called the program death molecule, but now it is known as suppression of the activation. Whenever it interacts with its ligand, which is PDL1, which is expressed either on antigen-presenting cells or any other cell like cancer cells. So whenever there is an interaction of PD1 and PDL1, it will suppress the activation of T cells. And it has been found that in cancers, many cancers, these two molecules are highly upregulated. And that's how they suppress the T cell activation, and the cytotoxic T lymphocyte are not able to kill the cancer, and that's how cancer survives. And based on these observations, very important discoveries came up when James Ellison from US, he proposed that if we use anti-CTLA4 antibodies in cancer, then these antibodies will go and bind to CTLA4 and will not, and that way CTLA4 will not interact with CD80, and then the CD80 is left free to interact with CD28, which passes on the activation signal, and that way the T cell get activated and it kills the cancer. And on the similar grounds, another scientist in Kyoto, Japan, Tasuku Honjo, he proposed that anti-PD1 antibodies or anti-PDL1 antibodies will also do similar process in cancer, because that will block the interaction of PD1 and PDL1, and that will lead to activation of the T cell, which will kill the cancer cells. So this has come up as a immunotherapy of cancer. And for these discoveries, these two scientists got Nobel Prize in Medicine, we all know, in 2018. And that immunotherapy has been approved by many regulatory authorities all over the world and is being used on cancers. And it is to some extent very, very, effective. But this effectiveness is not 100%. So why in all cancer, these immunotherapies, they don't work? There are two reasons. One is that there is a differential expression of these molecules in different cancers. Some cancers, they do not express such molecules. Some cancers, they do express very high quantities of these. So all those cancers which express PD-1 or CTLA-4, they will respond to the immunotherapy, whereas obviously that's the reason. Then the other part is that these two molecules basically act as at two different stages of T cell activation. While CTLA-4 acts in the priming stage when the antigen presenting cell is initially priming the T cell and it blocks the second signal. Whereas the PD-1 acts at the effector stage. It is at when the T cell has already activated and it is trying to perform its effector function, but the expression of PD-1 and PD-L1 blocks the activation, and that's how. So these two molecules act at different stages, and they may synergize. So it was thought that a combination of these two monoclonal antibodies, which can block both of them, may increase the efficacy of this. So while these thoughts were coming up, more research showed that besides these two molecules, there are many other checkpoint molecules which are expressed. And they include TIM3, LAG3, TIGIT, BTLA4, etc. And all of these have got different mechanisms to suppress the T cell functions. But all of these have independent way of suppressing T cell activation. And it is also now indicated by the recent research that besides cancer, even some infections also utilize this strategy to manipulate the expression of these checkpoint molecules and evade the immune activation. To understand the role of these checkpoint molecules in infections, in addition to cancer, I will quote some more study from our lab, which are very interesting. And one of these studies is still going on in my lab, as one of the PhD students is still working. This is regarding the HIV-TB co-infection. We know that HIV, Mycobacterium tuberculosis infection is the most common opportunistic infection in HIV patients. When the CD4 count goes below certain mark. So these people become immunocompromised, and most of opportunistic infections, they occur in these patients. And MTB infection is very common, and about 50% of HIV positive individuals, they basically get infected with that MTB, or the latent TB in more endemic countries. When there is a lot of people, they carry the latent tuberculosis infection, that gets activated. So that happens more often in African countries, African subcontinent, as well as Southeast Asian countries, where the TB is more endemic, including India. And we have a sizable population of HIV-TB co-infected individuals. And clinically, if we look at these individuals, they are also basically put on treatment. But if we look at the long-term profile of disease in HIV-TB co-infected individuals vis-a-vis HIV mono-infected individuals, it is apparent that the HIV-TB co-infected individuals, they progress to advanced stage of disease, which is called AIDS, much earlier, and they die, and the mortality rate is also high in this population. So the questions which we can ask is, why the HIV disease progresses faster in co-infected individuals? Are there any immune mechanisms modulated by Mycobacterium tuberculosis bug which help the virus to survive and keep replicating in these individuals, which is a host for co-infection? And if that is true, then what are those mechanisms which are playing the role? So while working on HIV-TB co-infected patients in our lab, these students showed that MTB modulates many immune functions, leading to dysregulated expression of many genes. And some of these mechanisms, they help the virus to survive and replicate faster. Then another student who showed that HIV basically evolves genetically in the HIV TB co-infected individuals much faster as it accumulates large number of uh, drug resistance mutations and other genetic alterations like additional we found that additional NF kappa B sites in the promoter region of the gene for this HIV patient. So that basically leads to genetic evolution of virus, which makes it more replication competent. So that was the observation when another student came up in the lab who proposed that let us see if the Mycobacterium tuberculosis manipulates with the expression of checkpoint molecules, because there are some papers in the literature where it has been shown that in the HIV patients who progress faster, they showed higher expression of one of these checkpoint molecules. So my student, she started looking at more than one, 4 or 5 checkpoint molecules and tried to see what is the expression level of these in the HIV patients' T cells. And she found that there was a significantly higher expression of these molecules in the cells from HIV TB co-infected individuals as compared to the HIV mono-infected individuals. And she also showed that this expression level is also correlated very nicely with the decline in CD4 count and higher viral, which both of these parameters, they indicate that the disease was progressing faster in these individuals. So altogether, this indicates the upregulation of these molecules leads to faster disease progression. So we can make some general conclusions here about today's podcast. We can safely say that in order to survive and propagate in our system, some pathogens, mainly viruses and cancers, they do interfere with the process of antigen presentation, and they downregulate the T cell activation process via one, that they downregulate the maturation of dendritic cells, causing missing second signal, leading to passing off a tolerizing signal. And we have seen that in HCV and HIV infections. And second reason is that they upregulate the expression of checkpoint molecules, which suppress the T cell activation process. And we have seen that in cancers, which has led to development of immunotherapy, which has revolutionized the treatment of cancer, and more efforts are being made to use combination therapy. And we also talked about that similar possibilities have been indicated in many infections situations, like I have shown you some data about HIV TB co-infections and chronic hepatitis. So I will close down here. I will encourage you guys to read our papers and some literature which is available. I have quoted here. And till the next episode, thank you very much and goodbye.
Thank you. This has been truly riveting, knowing how there are new prospects to such chronic infections and disease areas. Thank you. If we can look at just one or two questions that have come our way, please. How effective is immune checkpoint immunotherapy, and does it work for all the cancers?
Well, the answer is both yes and no. Yes, because it is highly effective in certain types of cancer, and even completely, eradicate the cancer in some individuals. The new studies have found that it depends on the expression level of these checkpoint molecules and their accessibility to the antibody. Sometimes in the solid tumors, the blood cannot reach that, and the antibodies do not have access to these cancer cells, which or the T cells, which are actually expressing these molecules. So that's why the response is low. And then we have also shown that there are multiple checkpoint molecules which can get expressed, and every checkpoint molecule has independent way of expression. So we have to think about a combination therapy rather than a monotherapy. So that's what happens.
And we have just one more. Uh, they're asking, generally, we see highly toxic side effects of all types of therapy for cancer. Are there any side effects of this immunotherapy also?
Yeah, yeah, sure. There are many serious side effects of this therapy also, which can be serious and even life-threatening. So they are caused because of overreactive immune response, leading to autoimmune reactions, but are usually manageable. Continuing research is focused on elucidating such mechanisms of action with the aim of improving the therapies and reducing the side effects. So, of course, there are some side effects there.
So you have your answers, listeners, and we will meet soon. Thank you, SKA.